I've always really loved nature and being out in nature. So I've always loved stopping and looking at everything and all the species I could see, like insects and flowers and everything. But I only really started getting into mycorrhizal fungi when I learned about how important mycorrhiza can be in providing nutrients to plants and how overlooked they are in our current agricultural system and in our crops. I'm Rose. I'm working on a PhD with both Imperial College London and Kew Gardens. A lot of people would look at what we do and it looks like we're washing dirt. But really, mycorrhizal fungi and roots are incredibly important. When we think about plants, obviously we know that plants have roots. Through their roots they take up nutrients that they can only get from the soil. But what a lot of people don't know is that most plants are actually using fungi to do this. 90 to 95% of plants need mycorrhizal fungi in order to take up those nutrients that they require from the soil. So understanding what type of fungi different plants are using, how those fungi behave in ecosystems, how we can make sure that those fungi keep surviving in our ecosystems when we have pollution and climate change and land use change all over the world is really important for keeping the plants in our ecosystems thriving. The word mycorrhiza means myco, which is fungi, and then rhiza, which means roots, so it's fungal roots. They do come above ground if they form mushrooms and fruiting bodies above ground. But really the best way to capture them kind of representatively in an ecosystem is when they're in association with roots. So you're looking for these roots in the soil. So you go out to the field, wherever you're studying, and you collect soil cores and you take them back to the lab. And then once you're back in the lab, you have to wash the soil away from the roots and then collect those roots and look at them down a microscope and look at what type of fungi might be living on the roots. My favourite step is definitely the field work. The ratio is like 1 day of field work to like a bit less than a week of lab work. And I wish it was the other way around.
Okay, so this is one of my samples that we've taken in the field from a corer. So we wash them. This is essential because basically it allows us to get a good sample of everything that's in the soil. So this is a technique we use to be able to pick out as many roots as we can. Depending on the soil type, this can either take a long time. This one's going to take bloody age. I'm Izzy, so I'm doing a PhD at Imperial. We're currently at Kew Gardens, the Garden Laboratory. I always had the prospect of doing research because I knew my dad had. He's always been in research, so I had an example of him, and I knew how passionate he was about his job. I've always been interested in mycelium, and I was particularly interested in how that affects carbon sequestration. So it's estimated that 35% of current carbon emissions are sequestered into the mycelium of fungi that live below ground. So obviously they're hugely important for climate change mitigation. The carbon that the trees take in from carbon dioxide, and they photosynthesize, and they take in carbon, turn it into sugar, and they feed it to the fungi, and the fungal structures below ground therefore make up a huge sink of carbon, so they stay in the soil, and it allows carbon from the atmosphere to be stored below ground for long term in fungi, and their importance has only recently been highlighted. So investigating how different communities might affect the below ground carbon, or how the below ground carbon might affect the communities, is a key question that's yet to be answered, really. So this is when we pick the roots out of the soil, and depending on what you're looking for, you would select different roots. So I've had a couple of projects where I'm looking at just a certain species of tree root, so I'd only take out that species. Tree roots of different species are quite distinct, usually, but it's still hard to tell if you haven't got a reference. So the staining allows us to identify fungal structures that are of interest because it shows you the traits of the fungus present. Fungi are so different depending on where they are in the environment. They can look so different, they can act so differently, they can perform different ecological roles. And so it's important for us to look at the morphology of the fungi in response to where it is and where it lives, which plants it's associated with, etc. and that can help determine the species present, the type of fungi present. But usually it's hard because they can look different in different environments. So that's why we take their DNA and then we sequence it and we see which fungi is which by their genetic makeup rather than their appearance, because that can be misleading. Each one of us have their own unique set of genetic codes, so DNA that we have. So everyone will have different DNA, but between different species, there's a certain parts of your DNA that will be replicated and similar between organisms that make up that species. So we can sequence that part of the DNA and we can compare it with loads of DNA that's been previously connected, and we can try and match it to a species. Yeah, so I think starting a PhD is initially very exciting, a bit daunting. When I first came here, I was completely overwhelmed.
I think there's always the challenge of having kind of like the feeling of imposter syndrome, which I think is really common from speaking to other people that do PhDs, because I think you always feel like you don't know enough to kind of make all the decisions that you need to make. And especially I think being surrounded by so many people who seem like they know exactly what they're doing, and like it can feel like they get through things faster than you and like understand things faster than you. It can feel sometimes like, what if I shouldn't be here? Like, what if I'm not good enough to do a PhD?
Unless you've done a master's in the same lab, you are in a completely different environment. You don't really know what you need to know yet. Just you facing what seems like a massive period of time and questions that you have no idea how you're going to answer. And obviously no one else is doing exactly the same thing as you. I think over time you gradually build your own knowledge base, and you've failed, so then you come back with better ideas, and you kind of learn that you can rely on yourself more than other people, and that makes it easier to decide things in your PhD.
I think I've also learned that I'm like maybe more resilient than I thought I was, like, and that I can get things done, and like I can, like, I don't know, figure out difficult things and like work around problems.
Yeah, looking back over time to the last, 6 months, you see how much you've actually learned. Like, I've actually learned so much. Like, I've completely chosen a different field. I can now think for myself. Like, I can now formulate ideas that I would never even have thought of. And yeah, that's quite cool.
One of the reasons I really wanted to do a PhD was I really wanted to kind of have that ownership over a project and have like the kind of control, and to be the person that could make decisions in a project. And I think that's one of the really nice things about a PhD that you don't get in other jobs. You feel like it is your project. You know, if you are ballsy enough, you can kind of move it in any way you want to. So yeah, you will end up by the end of the PhD hopefully being like an expert in that field, that very, very niche field.